Welcome to the Explainer. You know, if you reach way back to high school biology, you probably remember the nucleus being called the brain of the cell, right? Well, today, we're going to look at it through a much more accurate and honestly a way more fascinating lens. We're exploring the nucleus not just as some textbook organelle, but as a highly secure, incredibly dynamic information processing data center. This is the literal hub that stores the master code of life. It orchestrates the whole shebang, from DNA replication to genome organization and gene expression. So, how does a microscopic vault control literally everything about who you are? It's a pretty wild thought when you stop and think about it. Every single instruction needed for protein production, for cell division, development, even how your cells respond to the outside world, it is all locked inside this tiny, tiny structure. The stakes of nuclear biology are, well, they're astronomical. By understanding this space, we're essentially unlocking the secrets of genetics, biotechnology, and even cancer research. So let's step past the security perimeter, shall we? Okay, let's dive into this. At its core, the nucleus is a membrane-bound organelle that contains the vast majority of a cell's DNA, and it's the ultimate boss of gene expression. You're going to find this sophisticated command center mainly in eukaryotic organisms. So that's animals, meaning you and me, as well as plants, fungi, and protists. Having this dedicated vault is actually the defining feature that separates complex cellular life from much simpler organisms. Section 1. Inside the secure vault. All right, let's map out the actual physical architecture of our secure vault. First up, look at the nuclear envelope. Now, this isn't just a simple wall, it is a heavy-duty double membrane that physically separates and protects your highly sensitive DNA from the busy, honestly kind of chaotic cytoplasm outside. Embedded right into that envelope, we have the nuclear pores. Think of these as the strict gatekeepers. They tightly control the transport of RNA and proteins in and out. Then, floating in the internal fluid, we've got this dense region called the nucleolus. This is essentially an internal factory producing ribosomal RNA and actually assembling the ribosomes. And finally, giving support to this whole structure is the nuclear lamina. It's this protein network right underneath the envelope that maintains the shape of the vault and keeps everything perfectly stable. Section 2. Packaging the code of life. So now that we know what the vault actually looks like, we've got to talk about the logistics. I mean, how on earth do you fit a massive, sprawling library of master source code into a microscopic space without it turning into a tangled, unusable mess? To put the sheer scale of the data being archived here into perspective, just consider this number, 46. That's the number of chromosomes inside human cells, which are organized into 23 pairs. This DNA is ridiculously long. If you were to take the DNA from just one single cell and stretch it all the way out, it would be about 2 meters long. 2 meters. And yet, it somehow has to pack into a nucleus that is only micrometers wide. And this brilliantly illustrates the mind-blowing hierarchy of compaction required to make that happen. We start with the raw double helix DNA. To pack this down, the cell literally wraps the DNA around specialized proteins called histones, specifically types H2A, H2B, H3, and H4. This creates structures called nucleosomes. It's a lot like taking a really long thread and wrapping it around tiny spools. Next, these spools get tightly coiled together to form what's called chromatin. And finally, during cell division, that chromatin condenses even further into those highly visible chromosomes we just talked about. It is, without a doubt, an absolute masterclass in data compression. But here's the catch. The DNA isn't just sitting there in storage. It has to be actively read. And this is exactly how the vault organizes. Active code versus inactive code. Euchromatin is your loosely packed DNA. Because it's fairly open, the cellular machinery can get in there easily, which leads to highly active gene expression. On the flip side, heterochromatin is your tightly condensed, archived code. Gene expression here is vastly reduced. Some of it, like the constitutive heterochromatin found at the centromeres, is permanently turned off. But other parts, known as facultative heterochromatin, can actually switch back and forth between active and inactive states, all depending on what the cell needs in that exact moment. Section 3. Reading and shipping blueprints. Okay, so the code is perfectly packaged and organized. But how does the cell actually use these instructions to build things out in the cytoplasm, especially since it can't ever let the original precious DNA leave the vault? Well, the answer is transcription. Information moves from DNA to mRNA and finally to a protein. Because the DNA is strictly locked inside, the cell runs a highly specialized read-only data export process. Key players like RNA polymerase, promoters, and transcription factors team up to read the DNA and synthesize a messenger RNA, or mRNA, blueprint. But the cell doesn't just ship out that raw blueprint right away. No way. It has to be heavily processed first. It's almost like encrypting and stabilizing a file before you send it over a network. The newly produced RNA goes through 3 major modifications. First up, a 5' cap is added to the end to protect the RNA from getting degraded. Next, a poly-A tail is slapped onto the other end, which improves its stability and helps it travel. And finally, we have splicing. This is where the non-coding sections of the RNA, called introns, are basically cut out, and the crucial coding sections, the exons, get joined together. Only after all of that is the blueprint finally ready for export. And let me tell you, this export process is incredibly strict. Remember those nuclear pores we talked about earlier? They run a rigorous biometric protocol. For any molecule to even enter the nucleus, it has to possess a very specific protein code called a nuclear localization signal, or NLS. And to leave the nucleus, like our newly processed mRNA or a ribosome subunit, it absolutely must display a nuclear export signal, an NES. It's simple. If you don't have the right signal code, you do not get through the pore, period. Section 4, controlling the master switches. Up to this point, we've talked about storing, reading, and shipping the code out. But honestly, the most powerful function of this command center is deciding exactly which pieces of code to execute and when. This is the complex realm of gene regulation. So the crucial point is this. A neuron and a muscle cell contain the exact same DNA, but they express different genes. Just take a second to think about that. Every single cell in your body has the exact same master blueprint. The only reason a brain cell looks and acts so differently from, say, a bicep muscle cell is because the nucleus is acting as this ultimate master switchboard. It selectively activates and represses specific genes to determine the cell's entire identity. And one of the primary ways the nucleus pulls this off is through epigenetic regulation. You can kind of think of epigenetics as the software permissions or the system updates that control gene activity without ever actually changing the underlying base DNA sequence. There are 3 main mechanisms at play here. First, DNA methylation, which typically reduces or entirely silences gene expression. Then there's histone modification, things like acetylation or methylation, which can either turn the activity up or down depending on exactly where it happens. And lastly, chromatin remodeling, which physically changes how accessible the DNA itself is. These epigenetic layers are absolutely vital for life. Section 5, hacking the command center. But what happens when a system this complex and this critical experiences an error? And maybe more importantly, how is modern science learning to interface with it? When the biological vault is damaged, the real-world human stakes are incredibly severe. Nuclear abnormalities or chromosome mutations can lead directly to cancer, very often through the activation of oncogenes or the loss of crucial tumor suppressor genes. Glitches in the nuclear DNA are also the root cause of genetic disorders, things like cystic fibrosis or Huntington's disease. And on top of that, the natural aging process itself is tied closely to the nucleus. It's driven by the accumulation of DNA damage, altered gene regulation, and the shortening of telomeres at the very ends of our chromosomes. Now, what's really interesting about this slide is how we are finally pushing back. We aren't just observing the command center anymore. Biotechnology is literally allowing us to hack it. Through gene therapy and advanced genome editing tools like the CRISPR-Cas systems, we can now actually go in and correct disease-causing mutations right there in the nuclear DNA. Recombinant DNA technology lets us manipulate nuclear genes to produce life-saving therapeutic proteins and vaccines. And by studying how the nucleus regulates itself, we are making massive, game-changing leaps in stem cell biology and finding entirely new drug targets for cancer research. And the tools we have are only getting sharper. Looking ahead, the future research areas in nuclear biology honestly sound like science fiction, but they are happening right now in labs around the world. Scientists are successfully mapping the exact 3D organization of the genome. They're working on developing artificial chromosomes, they're refining precision genome editing for highly personalized medicine, and they're even using AI to predict genome regulation across millions of variables. We are literally decoding exactly how the central information processing system works. Which leaves us with this final, profound question. Now that we have the tools to rewrite the master code, how will we shape the future of human evolution? The cell nucleus isn't just some static storage space. It is the active control center of life itself. And as our understanding of it deepens, so too does our incredible ability to redefine what is biologically possible. Thank you so much for joining me on this journey inside the Cellular Command Center. Keep questioning, keep learning, and I'll catch you in the next explainer.